Can we be a little louder in the morning? Good morning. Yeah. How was your evening yesterday? Enjoyed it? Okay. So more or less everybody is kind of adjusted to the Indonesian time and you look all bright and cheerful. So it's nice. Yeah, yesterday we were missing one team. Today we have you on board. Okay, so we have with us another facilitator who's a super busy researcher, much on demand. So she could only arrive yesterday night and she's here with us today. I let Ari introduce herself before we start the session for the day. Ari.
Thank you very much, Farah. Hello everyone, my name is Ari Probandari. I'm a researcher in the Center for Tropical Medicine, Universitas Gadjah Mada, and I'm involved very much with Vara and Dorothy in the course about GSILP and IR. So nice to meeting all of you and looking forward for more dynamic discussion during this course. Thank you.
Thank you, Ari. Today, I think as they present, you may catch up with their topics, or maybe we can circulate it to you so then you can kind of get familiar with their topics. Ari is also very experienced and has a lot of practical knowledge to share. So it's very nice that we have her sooner than later. You have enjoyed the facilitation and the discussion, and that's one of the things to be appreciated about a face-to-face learning. And that's something that we miss on an online kind of a training. So we are happy that that is going good. And there were some remarks about the facility, and we have also brought it up. I hope the internet issue is solved. Looks like the internet is way beyond the hotel's capacity. It's like the telecom provider itself had some kind of a major setback yesterday. I'm sure it's sorted out. And with the air conditioning, there was also some remarks. We've also brought it up. I'm not sure if we would be able to sort it out, but then we have taken action. And as for the other remarks that you gave, we realized it yesterday morning that probably we should have let you know well ahead of time about preparation of this proposal, about coming prepared with a lot of questions. So we fully agree with all your feedback, and then we would try to adjust it in the upcoming courses. So thank you so much, and continue to give us this feedback, because we are only piloting. We have not yet reached the final stage of this blended program in terms of contents, methods, and its structure also. So we really look forward to your active participation. Okay? So having said that, do you have questions from yesterday that need to be clarified? Do we need to stop now to kind of take some questions, or you think we can handle them during the next session while you are going to be presenting? Any burning questions on your mind, or you went and you slept and then you woke up and something came into your mind that you wish to share with everybody? It happens, you know. If you have any such aspects, we could do it in the next session of presentation and discussion. So we start the day with a bit of recapitulation on the quality system. I've been using this word quality since yesterday morning, maybe 100 and odd times. So what do we mean when we say quality? Veni, there's some background disturbance there. I don't know what it is.
I'll answer.
Yes, please.
Adherence to a certain standard.
Fantastic, right? So, in normal life, who sets these standards? Who sets these standards? Whose standards are those? Your standards, my standards, or Maria's standards?
It's usually a consensus, or some studies have particular standards already. Like, for example, the JHRP is standard, or GCP is a standard.
Fantastic. So I just want to bring the fact that in daily life, we set our own standards. For me, a quality restaurant is something that provides me safe food. So then the next day, I'm actually here in the training and not suffering from any diarrheal disease. Somebody else may think, okay, my stomach can handle this food. I don't care about this. But for me, it's the ambience, Do I have nice hostess coming and serving me? How's the hospitality, right? So standards are also relative. So the first thing is, yes, quality is conforming to a set of standards. And in daily life, it's very relative to what we set as standards. But as rightly said, standards are preset in research. So if you're talking of a clinical trial, you say GCP is our standard. Now in this course, we agree GHRP is our standard. So you will see on the wall, Vinny has printed and she's put up the standards. So when you're going for your next exercise, you can just go have a look. Now these are the set of principles that we set for the GHRP, and this is what we are supposed to be complying when we refer to as standards. Yeah, so perfect. So it's conformance to a set of standards. And why do we need that? Because we need to be sure that all our processes are reliable, and therefore the data that we generate is credible, is repeatable, and is auditable. Now what does auditable mean? We just stop for a moment on this auditable because you will get the idea of why we talk so much about documentation and recording in research. What does auditable mean? Yes, Maria?
That you are able to reconstruct, to
Yes.
demonstrate every single...
Perfect. So you're able to reconstruct, to say who did this? What was the procedure that was used to do this? What was the method that was used? What is the equipment? When was it done? You will be able to kind of attribute all elements to this one particular data. So you can kind of reconstruct the whole study, right? And that's really very important. I think this has been also brought up in yesterday's discussion, how important it is for us to have this reconstruction, right? So we need to be able to show that this data, whether it's generated by us or by another team, is like repeatable and reproducible. We don't get results which is very different from what others get. And that's why quality, we are again and again enforcing not just quality of the data, quality of the process, and then quality of the data as well. Okay? So, and why do we need it? Because we have to have valid and meaningful conclusions. We are going to make choices. We are going to be coming up with policies and recommendations. So if the data is garbage, then the output that we get in terms of report and policies is again garbage. So we need to ensure that it's quality in and quality out. Okay? So how do we do that? We won't go into all the details, and that's exactly what we are trying to do. Have a very nicely written protocol, have clearly written procedures, ensure your research team follows all these procedures, and that's what we are talking about again and again. I leave this slide because the whole idea is that whether you did it or somebody else did it, we can have replicable, reproducible data in the same way that we did it, right? Okay, so now little more definitions. What's quality assurance? No, not Maria. You facilitate this course. Let's give a chance to people who are naive to GHRP. We use a lot of terminologies in our day-to-day life. I mean, it's not a strict definition, but are we understanding what we mean by quality assurance? If there's nobody, then I give it to Maria. Anybody wants to attempt? Okay, go ahead, Maria.
So it has to do with all the set of activities that you organize beforehand, so your processes are according to the quality standards. Mm-hmm. For example, when you develop the SOPs, when you develop a system that ensures that everything is going to be according to the standards that are set.
Perfect, right? What you're doing is you're trying to assure that all that you're going to do is going to fulfill requirements, namely the GHRP standards. So, meaning to say, in simple terms, you have the right person, you have the right facility, the right equipment, everything, right? This is what you're trying to do up front. Now, does that mean, in my research, I've got the best protocol, I've got the best SOP, the best team and best equipment. Can I just sit back and relax and then things will happen on its own? That's what happens most of the times. We write a very nice protocol, it gets approved by the IRB, we kind of get a quality stamp, We write the SOPs, we train people and say, fantastic, we have done a great job. And then comes a lot of problems later on, right? So can we sit back and relax? So what do we do? If we don't sit back and relax, what do we do?
You supervise and monitor what is going on.
Yeah, right? So we see that we need to have verification, process of check. Don't worry about that perfect definition that is being given. So basically what we need to do is keep supervising our processes, keep monitoring our data to be sure that everything is up to our expectations. And this brings us to this quality management system, right? So the quality management system is like the entire system about your research. It's about where, what kind of facility, who are the people, what is their training and qualification, what are the kind of equipment and materials that you're going to be making use of, what are the documents and records, your different processes, and it also includes this review and checking. Do you have systems to check your people, to check your equipment, to check your documents and records? So this whole thing is what is referred to as the quality management system. So now let's be honest and say how many of us in our research implement a quality management system? Depends, yes. Give your experience, Maria.
Because it depends. For example, we are doing a clinical trial that you
see?
can't escape it.
See that? So this is what happens in reality. So a clinical trial demands, GCP demands that you have this kind of quality checks. But in other research, we generally don't do. But yesterday we started by saying, in all our research, we have human participants. We are generating data. Then why we don't focus on quality management and we say we do it only for clinical trials? You have another experience? I thought you also wanted to share something.
We are
Yeah.
required to adhere
Yeah,
to QMS
right?
because of ISO.
Yeah, so can you turn your, I'm not yet familiar with some of your names. Yeah, right? So we always have this tendency, let's be regulated. Let somebody tell us to do something and then we do it. But we have to keep in mind, and this is the whole idea why TDR came up with this good health research practices, to emphasize how important it is for quality and ethics in any research involving human participants. So we need to set up a quality management system. So basically we use the same risk-based approach. We can't be checking everything, right? That will involve, again, a lot of effort and a lot of money. So again, I look at what are my key critical process, what are the key critical data. I may have some data related to recruitment, inclusion, exclusion criteria. I may have some data required to my endpoints. So I consider them as key critical and then I focus my review process on that, right? So anything that might impact participant safety and data quality. I look at the risk, and again the same things that we did yesterday. So if this data is critical, if this process is critical, how do I ensure I have quality mechanisms built into it to be sure that the data that I get conforms to my expected standards? So the process is basically the same. And you see this risk-based approach we will be using in all the modules that, all the practical activities that you are doing. So when we talk about this verification process, what exactly are we doing? We already set some standards, so we now have to measure whether what we do is in accordance with our standards. So you wrote an SOP for a given procedure. We need to verify that a person is actually following that SOP. And then see if he is not following, what kind of action you need to take. So basically, you set standards, you measure to see whether you're following the standards. Whatever data you get, you kind of analyze, and then you act. This is a very, very simple process. And we have some standard terminologies when it comes to research. Like Phyllis said, what we call as monitoring is something that your team will be doing regularly, systematically, and continuously, from before your research until end of the research. And how much of it you do, what you do, is a risk-based approach. Sometimes you may have multi-centric trials or multi-country trials. hugely funded by a third party, an international agency, and they would like to come and audit some sites. Maybe it's just a regular one, or maybe they find issues in a given site and they would like to. So an audit is usually done by an independent agency, whereas this is more like your quality control, which is built into your process. And sometimes you can also be inspected. It's not just clinical trials. Anybody doing research. There's something out in the newspaper about, let's say, I think they had this problem even with their Wolbachia study. So a journalist wrote something on the newspaper, and then the governmental authorities wanted to come and see if everything is going on fine or no. So inspection can also happen, not necessarily only for clinical trials, right? So what do they evaluate? They're going to look at your documents, your processes, data, anything for that matter. And what approach are they going to use? Is again risk-based approach. Right? Nobody is going to look at 100% of your processes and data. They're only going to look at certain components of it. Right? Which means that your documents should be very nicely made and self-explanatory. Many times we don't understand your data collection tool. It needs explanation. So you need to design your tools and forms in a manner that not only you understand, anybody who looks at your documents and records are also able to understand. So that's the key when you design your tools. So what happens? When you verify, what happens? They pick up problems. Maybe your informed consent form, there are no signatures. Or maybe it's an illiterate participant and then the witness is also illiterate. How many times we see a thumbprint that's also supported by another thumbprint? So what kind of an informed consent is it? Is it valid? No, it's not valid, right? So the process of evaluation picks up all these kind of errors. So what do you as a research team do with all these errors? You need to use some tools to see where you act and how you act. So there are some tools that you can make use of. One of them is called as a Pareto chart. So I have 25 errors. I just categorize my errors and make it like a histogram kind of a picture. And I say, okay, this is the maximum error and this is where I need to address, right? Because what did we say in our risk-based approach? You may have risks that have low likelihood and low impact, and I cannot act on them. I need to work on those that have high likelihood and high impact, right? So I won't be wasting my time just looking at this one thing, but I need to prioritize and act on this. So you can use something called as a Pareto chart to say how you prioritize. Or you have another example, this is called as an Ishikawa. This is nothing but a root cause analysis. I find issues in my informed consent. What's the problem? Is the management problem? Because you're not paying your staff well enough. They are unhappy and they want to show their unhappiness. So they do it like that. So it doesn't have to be any problem with your training, nothing. It could be a management issue. Or it could be that people have not been trained. Or maybe your SOP is not good enough. Or maybe your materials and equipments are not good enough. So you can use this kind of an analysis to say, why is this problem happening? And then you can try and address. And we use a very standard terminology called as corrective action and preventive action. So whenever you find an error, you need to correct it, but you also need to prevent it from happening. So I see problems in informed consent, right? I see incomplete informed consent. No, I think this is a different one. I don't know why. Okay, I think I'm using a different presentation and I'm not version controlling. Anyway, so what happened? Okay, so if I see problems with informed consent, I have to see whether it's related to the SOP, then I correct my SOP, whether it's related to the training, whether it's related to people, whatever, and then address it accordingly, right? So we do, for every non-conformance that's identified, we not only correct, we also try to prevent these errors from happening. So, to conclude, When you have a quality management system, it does not mean that you will work in an environment where there will be no errors. We are all human beings. Technology fails. Like yesterday we saw how seriously technology failed, right? 2 of their laptops got crashed yesterday because of a major electrical problem. So technology fails, human errors can happen. So by having a quality management system, we are not assuring that we are completely error-free. We are only assuring that as and when something goes wrong, we are able to pick it up and then we are able to correct it, right? So that's why the corrective and preventive action becomes very, very important. So you will be able to pick up and keep controlling them. So the very last is this one, what we call as quality improvement. We never reach a stage where we can say I'm 100% on quality, right? You have your protocol, your SOPs, your training, you do your research, you keep monitoring, you find problems and then you act. But you don't stop there. You continue working, continue picking up errors, and so that's why we say quality improvement is key, right? We're doing, this university organizes training very well. It's one of those RTCs that really organizes training very well. Yesterday we wanted to have a nice fun session, but then that baby shark dance couldn't be shown to you. So we kind of come up with some alternative. It's okay. But if you imagine that we're research data, something goes wrong, and then I use an alternate procedure to generate data. So I have an apple, I have an orange. But I compare my apples and oranges as though they are equal and then I analyze. So how important it is for us to look at process and data quality, right? So we won't take much of your time. If you have any questions or if you disagree and say, no, we don't want to have quality, then it's time to say bye-bye and leave the course. But if you think, no, we need to have quality, we continue to go ahead with our activities. Yes, Maria.
Can you go again to the Pareto chart? Pareto chart, because first time I see it, so I would like to understand it.
Perfect. Okay. So I'm sorry I put an inappropriate example of the Pareto chart. Say, for example, you have somebody within your team picking up problems. And you see informed consent errors, you have 25. And then let's say this is about some equipment that you are making use of and that failed so many times, right? So what you're doing is, I did a monitoring of my entire system and I found 5 different kinds of issues. Issue number 1 happened 25 times. Issue number 5 happened only 1 time, right? Which one is the one that I need to act on first? Or it could be sometimes like the one that happened yesterday, the dance didn't work. In a training program like this, if the dance didn't work, it's still okay. But if something went wrong and we couldn't project, we couldn't conduct the training as we proposed to, that's for us a very key critical process in this training program. Even if that was only 1 error, I would prioritize that 1 error because that's the most important process, right? But for me to visualize and see how many times a given problem has occurred. And then I look at it and I say, okay, this has occurred 25 times. There's something seriously wrong there. Maybe the way I train people itself is not good. Maybe the SOP is not good. I need to work on that first, right? So it helps you prioritize, it helps you visualize what kind of problems you have. Now when I want to start working, okay, I have a problem with informed consent. Then I use the next one to say, is it because of a management issue? Is it because of people's attitude? Is it because the informed consent form is not well written? So then you start going into what exactly could be the root cause. And once you identify the root cause, you come up with this corrective action and preventive action. There are lots of these kind of tools. They call 5M tool. Man, machine, management, money, something like that. So lots of tools are there. But the simpler you keep is also the better. So that way Pareto is like kind of gives you a visual appearance, and then you can kind of identify, prioritize, and then go ahead to do this root cause analysis. Right? Okay. So now I hand over to Doni.
Thank you very much. Good morning, everyone. Have a good breakfast? Good. We will again ask you to think and clarify what doubts, questions, clarification, or inputs from the online phase. And I will, this is for the module 3 and module 4, right? And here we have several units and also classroom. In module 3, we have this study teams and sites, data management, research oversight. And module 4 is mostly on the data analysis, monitoring, evaluation, and reporting dissemination. So Dorothy has already explained yesterday how it works in the classroom and everything. So I would like you to, again, hopefully, I don't know, I knew that you arrived late last night, right? Around 10 or 11? 10, and you still have your jet lag. And I don't know whether this morning you can wake up quite early, but if there's any chance that you probably see the online modules, whether you have some things that you can highlight for the improvement. Okay, so what you think from these units is just in general, you think is the most difficult to understand, the units or classroom? You have some ideas now, or you remember something? Mari, Amachi?
Okay. Well, in general, I found in these two modules, I like the way there are a lot of charts and tables, and I think it's useful to remember and to illustrate the concepts. Again, maybe I feel that there are some big difference between the presenters, the facilitators, that are, so sometimes you kind of have difficulties to engage in the same way with some of others. I really like in this part since we were studying about data management and data analysis and data management, I like the way it is structured and organized to differentiate quantitative and qualitative. It was very clear. and very well structured. And I forgot yesterday to tell you that I noticed that in some videos there is the possibility to activate the subtitles, but not in all. I don't know, the English subtitles, of course, but yeah, I couldn't find for some of the videos, because sometimes it is helpful because, the different accents and those kind of things. No, I think that's it.
Okay. Thank you. Ellis.
And because Machi mentioned that I was going to the study team and sites again in reference to the tables and the charts. So they were very helpful. However, when we transition from one to the other, it's as if we've not said the previous.
Yeah. The linkage.
Exactly.
Yes.
A key example is the OBS, after which we move to the responsibility allocation. It's like, so when do I use the OBS? How is it related to the responsibility? And I think that presentation was by Doni. Like, we had all these charts, all this flow things that is helping us to understand, but the connections.
Okay.
Yeah.
Yeah, I think that's what we are lacking because of. It's good that we can sit together and identify how to link it from. We will do that in the next session.
But I think it worked for the data management.
Okay.
Like, for instance, the 3 C's that you presented was very clear, and it was connected to the previous. So there are examples that I think were very good that you can just follow through for the others that weren't clear.
Okay.
Thanks.
Thank you very much. Any other highlights?
Any specific
Yeah.
questions related to the contents?
Yeah.
Because we are now looking at you applying all this to create your own data management plan. So we're really looking at, do you have any questions? Were you able to understand the content?
In relation to that, with the project data and the research data, I really appreciate that this course has project evaluation component in it, but in some places I think we can be more clear about what to do with the project data, because when you're in monitoring and evaluation, you tend to generate a lot of data, because if it's your research data, it's easy to manage, especially when there's a system in the institute. But with project data, sometimes it's hard to know whether you have to collect this particular data for monitoring purposes or for evaluation purposes. So as project coordinators, we would appreciate if there are more contents on, or a more systematic
Content on that, addressing that.
Addressing
Okay.
how to manage project data.
Okay. Because in those we discussed that some data should be kept, some data you can discard it, and then particularly the study data, but for project data, and that's also related to the essential documents, things that needs to be there to reconstruct. Yes, I think we can add more on that later. Farah, you want to add something?
You also missed a little bit of the session yesterday where we wanted to make this distinction. And like Phyllis immediately brought up, she said, oh, but I am a scientist, I really don't have the time so much for project management. And what we want you to really look at is that there are 2 components to it. And if you need to have your research successful, you need to manage your project. How you do it is dependent on the setting, dependent on the type of the research. Not in all you may need to have this, very clear separation and all that. But in multi-country, multi-site, heavily funded projects, maybe you need to have a clear distinction. If TDR gives you just a $25,000, you can just manage the whole thing yourself. So you also need to make a choice when you would want to make this demarcation, when would you want to spend more on project management, when you would want to do less. But what we want you to look at is that there are two distinct entities. Don't forget that. Who plays the role, whether it's one man, one woman, or is multiple people, is your choice. But don't forget, that's the message we want to kind of deliver. We found that, we picked it up, and so yesterday we kind of had a quick session on risk assessment and we made this specific. You missed it, but we will definitely look at it and improve on that.
Especially because you are a project coordinator.
Yeah, we're project
And that's
coordinators.
something that, yeah, thank you very much.
Questions on the contents.
Yes.
Really looking at the, because you have now two exercises for today. One is going to be on coming up with your data management plan, and in the afternoon you're going to be doing the study project management. So if you have questions on those, then Ari and Doni will be able to clarify.
Yeah.
Because we also have a session to this evening where we are going to do a collective feedback on the way the online and the offline worked, and we have some specific bullet points there. So for now, questions on contents. Otherwise, it's like super delivered contents. They can go and start their practical activity. No more questions? Yeah.
HR.
Continuing what Pak Doni said about the data, the storage of the data management, is it any difference between GHRP and then GCP? Because that I heard in GCP that we need to keep the data for 15 years and also the essential document for 2 years. But how about if we conduct the research in human participation?
It depends. It depends. I mean, it depends on several regulatory, what you call the requirements that you have to follow. Some regulatory requirements, like a GCP, it has a very long, because in clinical trial you might need to have a post-market, and then you found something, you have to go back. But in public health research and other, it's not that. It depends. I think best way is either to follow the funders or the IRB. You have a additional thought?
Institutional policies.
Yeah.
So whichever is the longer.
Yeah.
So if IRB says 5 years, but your institute says 8 years, then you better follow which is the longest of them. Yeah.
Yeah. Thank you very much. Okay.
I guess I just need some examples because this is quite a dilemma for a resource constrained setting. It's stated somewhere that your backup should be, there's some kilometers where your backup of like electronic files should be housed somewhere else. But maybe I just want to get some suggestions how you manage that, because to be honest, we don't know how to do it in our institute. But in the case that the building burns down, all our data are in the same building actually. So we were thinking of putting it in the cloud, but it's too expensive for now. I'm hoping that that would change in the near future, but may I know how the others do it?
Again, it depends. I mean, putting the backup in other location is just, again, backup. If something happens with the location when you store now, you have somewhere else safe. For example, if there is a fires, earthquakes, floods, in those areas where you put your data, then your backup is still safe. That's the main idea. But I think nowadays, I mean, cloud is an alternative, but there's also other issues with securities, and then with the legal requirements, because in some countries, they said that all identifiable data should be stored in countries. And while in clouds, we don't know where the server is, right? So that's something can be problematic. But I think if, I perhaps Diljit know better, the price for cloud services is now quite cheap. For example, if you use Amazon, it's very cheap. It's just to store. But if you have require advanced management system, then it a bit quite expensive. Oops. Sorry. Yeah. Yeah, I think we need to.
I think you use this side. This is less wide.
It's a risk assessment. They have to put. Okay. Is it quite clear, Farah? You want to add something?
Yeah, if you're looking at paper data, then we also have now repositories like your bank locker kind of system. So there are also third-party facilities where you can safely store. Of course, you've got to pay a huge price for all these things. And yeah, and OECD has got a lot of very nice guidelines because they are very crazy about data security and privacy and confidentiality. You can look up to get more insight what they do and how they do. You don't have to 100% apply, but you will get better concepts because they have very clear guidelines on this. Yeah.
Okay.
Yeah, and so in relation to the more examples, I think that for me is the practicality. Putting it in your protocol to satisfy IRB is one thing, and then you come back and you realize that all that you said is really difficult for you to practice. In that case, the gaps, how do you manage it, and then make sure that at least whatever you are writing is credible and authentic that you can follow?
Yeah, I think before you write, you really do explore and be sure what you can. And if you cannot follow certain guidelines, be very honest with whoever your funder is and say, this is our limitation and this is all that we could do about it. And is that okay? So I would rather say, write what you would do. Don't write more than what you actually can handle. And that practicality is what we always keep talking in our course. If you see, planning is about 75% of the course. So you need to really understand the contextual situation and draw your limits and say this is all that we will be able to do. That's very, very important.
I just wanted to add to that. So, but it is a practical problem that very often we say something and with excellent intent, but we are not able to follow it through. In which case, I would also suggest going back to your IEC and telling them that this is what I said, but this is not what I'm able to do. Can you help me think of a solution? And IRBs appreciate that. So they're not only regulatory, they're also, their oversight. They're supposed to help you through this process and help you find a solution. So if it's an absolute practical problem, I think you can go back to your IEC and find local solutions for
True,
it.
that's I think also our case. Just recently we found in our study that a lot of children without proper LAR, we cannot recruit it. So we asked the IRP how we can somewhat handle with the same, also within the legal system, and then come up with recommendations. So I think always consult with IRB will be a best way to do.
Last one.
No, we still have 5.
So the other question is, so again with the practical aspect, if I'm planning for data management, where do I start? What should I think of first? And in moving through the processes, what is next? Is my design the most important thing to look at? Is the outputs the most important thing to look at? Is just picking some quality assurance processes what I'm supposed to follow? Just share experiences here.
You want to start, Farah? Okay. Well, in our experience that we start to think the data management when we start to write the protocol. Try to think what kind of, well, start from what the outcome you want to have first in your study. And then thinking backwards, and then try to see what kind of data you want to collect, from which source, and then you have to think through the data flows, because data can be from different sources, and you need to reconcile or put it together, and those flows is really important to be able to reconcile. So think that through, and then put it in the data management plan. Those.
If I can just add to that
yeah.
also and say from practical experience how, so definitely design is first, then you come to your methods, you know what methods, you already know what outcomes. Then you know what tools you're going to use to be able to collect that particular data. Then you're thinking through the data flow. I think that really helps us personally, is to think the data flow through. So you know who's collecting it, where is it going to go, where is it going to be stored, and you're thinking through processes at that point. Then you're putting in quality assurance mechanisms and you're thinking through monitoring. So I think it's in that order that you think it through.
Thank you very much. Maria.
Maybe something that, based on the exercise from yesterday, I think is also for us is very helpful to make the dummy tables. Because then we know how the data we're going to present it, and then it's a good exercise because then we realize that maybe there are some things that we are missing from how the data is going to be presented. So we imagine the paper, how it would look like.
Yeah.
So that's very helpful. Maybe we can emphasize a little bit more on that.
True. And especially true with the IR, because we have to think first what are the audience and what kind of information they want to understand from our findings. And we go back to what kind of information, data, and how can we collect it, those things. So having dummy tables, end result that you want to present to the what audience, it helps also. Yeah.
I think you can kind of map your entire start to end, and then that pretty much covers like everything, and then that'll be very good. But that takes a lot of time and effort.Maybe it requires a team as well. It's not going to be like a one-man thing, and that's also the reason why we always see 3 people come together for a course like this. So then you come with different ideas, and then you can kind of say, okay, this is my starting point, this is how my table is going to look like in the end, these are in between, and how I do that. Yeah, but you will go back and forth. There's no one-way movement. You start from behind, go, then you will be going back and forth till you reach a certain shape.
Since we're just talking about flow, and all of you, I think, are more from the quantitative background, so it's easy to think of it in that fashion, but a real practical problem is with qualitative research and the iterative cycles with thinking through, even changing question guides. And that really becomes quite a challenge in keeping track of everything. But I think the same rigor has to be applied there as well. Just wanted to add that.
Okay, you press the button before. You want to say something?
It was answered during the conversation. Thank you.
Any last burning clarification questions?
With regards to the evaluation, we are doing an impact evaluation of a certain thing, but it does not include, it is not particularly, it's an isolated study. It's an evaluation study. So when it comes to that, it was a little difficult for me to apply the module on monitoring and evaluation because I'm already looking at the impact. So I think the unit was just more on linking the outcome to the research objective. So I think I just need clarification on the level of which we evaluate, for example, research data.
I think here also when we say evaluation of the project. So it's not only the type of study that we do as evaluation type study, but this is evaluation for the project. So how can we measure that we achieve what we want to achieve from the project, and also those kind of things? Dorothy or Farah, you want to add something?
Yeah.
Okay.
I think you really don't have to worry about impact evaluation. Yeah, I think it's more like, how did your project go? What lessons did you learn? How could you do it better? It's more like a real learning experience and for betterment for a later point in time. But it's not the kind of evaluation that you do of big programs and things like that. That's not the intent. And we also see that that module went a little beyond what we wanted to be covered, so we're going to tone down that module. Thanks for
Okay.
bringing it up.
Yeah, it's very
but
clear now.
we realized that this is going to make people very uncomfortable, and that's not our interest. So we are going to tone down. We're looking at it more like a quality management, saying, okay, this is the resource, this is what I had, this is what I intended to achieve. Did I achieve what I wanted to? Could I have done better? It's more like a learning experience, and it's not looking at that big morbidity, mortality, those kind of public health things. So we are going to modify that one topic, and thanks for bringing it up.
Yeah, I think it really
Yeah.
has to be delineated.
Definitely,
'Cause-
not to worry. Sure.
Yeah, because monitoring and evaluation for us is actually a new, all the jargons are quite new. So impact evaluation is another thing, but
No.
is now being more commonly required. So yeah, that would be
Yeah.
helpful.
And now and then we have to differentiate between evaluation study and evaluation of a project. And by this GSRP, it's more on the research project that we want to evaluate.
At it from a quality management system. It's like quality improvement. What could have gone wrong? What could have gone right? How could I have done it better? That's the kind of focus. And so we have already noted it down that that module needs to be greatly simplified, and we will do that. Yeah.
Thank you very much. So next, what will the... So now we have a group work, data management. The exercise, you have the template, Diljit? So that Diljit, you can explain it better, Diljit. So we work on that, Google slide. Then you don't need to, you just work directly from your computer. You don't have to go back and forth and walk through those entirely. So, but you can, I think you can explain better. Yeah, yeah. So that you work on only on those files and your computer. You don't have to do anything else. And yeah, good.
So if you just give me the last version that you're working on, the same thing can be converted, and I will share you a link in which you can work.
Just so that there is continuity with the
Continuity what you have already done.
put all of it into a live document. Are they the ones? I don't think we have everybody's. I think we don't have the visibility, but we have the.
Yeah,
Maybe you better send it once again to them.
yeah, that's fine.
So all of you could email the last presentation that you made with the updated slides. Send it to Diljit.
Okay. Yeah, I'll give you my email.
Maybe write it on the board.
Here she can assign a patient to the follow-up of the treatment. Or they have developers. And they agree to give us a copy of that. We don't know yet what they are doing. Okay. Hello. Okay, so this is what we did. So we made like a small dummy table for, it's not extensive, so we summarize like demographic data, clinical data, etc. And then, yes, so for the effectiveness, then we need the treatment, the adherence to treatment, therapeutic response, and a table for safety. So this is like a very big summary of what we want to present for that specific aim. And then for the data we want to collect, then we have some methods. So for example, as based on our prior experience, so it's not feasible for the community health volunteers to go every day to monitor the treatment. Plus, if it's miltefosine, it can be 3 times a day, so that's just not going to happen. So we need to have some self-reported data collection. So we have the patient's diary, where they just report the symptoms and they report the doses that they are getting. And then we have like an SOP, some sort of training, how to use, to explain to the patient how to use it. Regarding compliance to treatment, so these are all, I mean, they are most, I think almost all quantitative that type of data collection. And then these are the data we're going to collect. We are going to collect both in apps or paper forms, so one of those depends on the feasibility. And we have an SOP, and we have a training plan for the community health volunteers. That was also a requirement from our IRB. So we had to have a training plan for them in ethics, in study procedures, so that is already in place. I think we will have to update it when we get new procedures or things, but this that exists already. For the adverse events, then we have 2 different, because I mean, we will collect data in the app or paper form for the non-serious adverse events, but once a serious adverse event is identified, then it has to be referred immediately to the healthcare facility. So then there is a report specific for serious adverse events. It's a national sort of document. So then there are 2 SOPs, one is for the healthcare volunteers and the one for the healthcare providers, so how to report the serious adverse events. For regarding treatment response is the same, so one for the field workers and one for the healthcare providers. Some of them already exist. I mean, for example, how to measure the lesions, how to measure all that exists already in the institution, so just to give them training. For the Leishmania species, so then we have SOPs for data collection of that variable, and we also have institutional SOPs that how to identify the species, so that we don't need to create for this particular project. Also for the demographic data that is collected via the apps or the paper forms, then we have the same sort of training. For the clinical characteristics, so we had a discussion yesterday how to handle 3 different sites where they have different paper records. So we came with an agreement that we need some sort of participant record that we ensure that all the data is being collected as we wanted to. The clinical records and the CRFs, and then a training for the healthcare providers in each site, so how to measure the lesions, the important variables that we want to measure, and also for how to take the photographs. There's also an SOP for that, and that will be collected via the apps in the field, but also a photo is taken in each clinical visit. So for the data management plan, so we have this flow chart. So let's see, so for the first aim, which is, so the green colors represent databases. I don't know what colors is this one, sort of yellow one are electronic data capture sort of. And the papers are in orange. And then we have, for one of the aims, if you remember, it's a lot of surveillance data, so these are already databases. So we need some identification of what data to extract from those databases. And for the lab reports, we expect them to be probably in paper, because this is just basically going to the facilities and checking. So then there will be a eCRF that will be filled by the field coordinator. And this is all going to the data center in our main facility in CDAIM. So for the aims 1 and 3, which is part of under ascertainment, and aim 3, which is effectiveness of treatment, so we have a community-based surveillance strategy. So then we will have paper forms that will reach the field coordinators, and also electronic CRFs, the apps basically. This is a form of electronic data capture. And all these centralized in a software in the computer where he can administer all the data that is coming, so the field coordinator can visualize whatever is two ways. So all this information is going to a data center, which is a third-party data center. It's not ours because it's for the developers of the app. So they agreed to give us I don't remember exactly, but they will periodically send us a copy of the database for our analysis that will be clean in the data center in CDAIM. And this and that are going to be part of the database for analysis. And the stars are the points where the monitor will be checking, because it was for the IRB and also for the funding to have a monitor, because it's a very complex project. So this is more or less our data management plan.
Any comments, questions, suggestions?
No, I just wanted clarification. The stars that you've put, can you just elaborate a little bit what you were thinking of the monitor will do?
Okay, so the monitoring plan is being developed, so it's that we don't have the final version, but what she will be checking is, it depends on the kind of data the primary data is coming from. For example, for this, what we need her to check if that the data extraction is working as intended. That is like basically supervising that the process is being done correctly. Also for the eCRFs, that is easier to check because we can have a primary source data. We have a source data to compare against, right? So she can, basically what she does is that she checks against the source data if it's the record is being correct. And for the paper forms is checking that the procedure is held correctly. And if the data is complete, so all the variables are being filled, because this will be the primary source of data. So whatever is collected, so it's more checking the process, because we don't have anything to compare to what happened. Yeah, we don't have anything to compare with. For the clinical records, that is also, let's say, verification that is, it has been done. But for these points is basically checking that they are being filled correctly, then it's more verifying the process, the training, because this would be the primary, the source data. And this, that it's transferring all the information that we need for this from the third party to our data center.
So just to add to some other things that you may want to think is, how many is she going to look at? Because I don't think it will be possible to look at every single one. So that percentage, usually 10% is acceptable, but that's something to think through how many you want. For the community-based exercise, that will be the questionnaires that you will do. Another way of doing it is to get another person to fill in the same form from the same household. And then you you verify between the two. So those are some ways in which people do quality checks there. Another thing is, of course, transcribing. So entry from the form into the database. That's another point you want to probably look at to make sure there are no errors in putting into the database.
Some ethical concerns about this third party having access to all this data, particularly you're talking about taking pictures of lesions and everything. So is there any way that can be bypassed, or what kind of agreements that you have with them, because that's for me kind of a big concern.
Well, they are part of the, I don't know how to say it. So they are part of the team, so the funder knows that they are part of the, I mean, the ones who developed the app, so we have collaborated before, but they have their own servers and their own things, and they also have information from other projects, their own projects there, so they just don't want us to get access to their data. That's why the agreement was that the data that is for these specific projects will be transferred to our database. But there are agreements between the two institutions, because ICESI is like our partner university, so we are not the same, but we are very linked. I don't know how to say it. But they are not part of the study personnel, but they are linked as a party inside of the proposal.
I think your agreement clearly needs to spell out what their rights and obligations are, that they only store, that they cannot further use this data. Okay. And if they further use that data, then it has to be only in consultation with you. I mean, imagine if they have data from multiple sites. It's like big data. Now I have data from multiple sites and it's accessible to me. What can I do? They could still say I anonymize everything, and then they could kind of do a meta-analysis of all your things there. So you need to draw a line of what they can and what they cannot do with the data that they have access to. This is stemming from our big data concerns these days.
Yeah, thank you very much. We'll check that.
Okay, I think there should be some kind of governance between both parties. For example, in terms of level of access, what kind of access they can have for that, because then perhaps they can have one dedicated person that can be, but you need to know who have access and to what level of access they have through your database, particularly the database, because they can develop apps, but it should be secure database, and only certain personnel can have access to that with the different level of access.
Comment with Donnie, I think the one that responsible for managing the overall data should be there. So that's the PI, for instance. The PI could not access by him or herself to the data. So every part of the data management should be passed through the data manager. That's very important things. Even the PI should be past the data manager.
I do not understand.
Okay, so because you have many parts of your data, then there should be someone that is responsible for monitoring the flow of the data, the data that come in the database, as well as the data extraction to the investigators. So it's through that data manager, then the researcher should pass through. When for instance, the researcher need the data, they could not access by him or herself. So it should be the data manager who will extract the data and give the data to the researchers.
Oh, okay.
It's like a fort. You have the data and there's a fort around that data. If anybody needs to pass through that fort, including the PI, there must be some checks and mechanisms that you have some user ID, password, not even user ID, password. You get something like a one-time password that you can only use now, and that's it. And then this leads to an audit trail of who accessed, when it was accessed. So we know completely at any point in time something happens, we know that only authorized people have access, right? So that's the kind of mechanism. How did your IRB perceive this? Did your IRB raise questions on this, or were they okay with it?
They are okay with it. Maybe I'm not so sure they have thought through the complexity of that. But we have worked before. So it's for this with this partner. I put a third party, but it's actually a partner of us. We have worked before. So it's not the first project. So the development of the apps was made between CDAIM and them. It's because we just don't have the IT capacity to develop it ourselves. So initially it was not, I don't know, I will have to think about it. And regarding that other point about so this is part of a program, so there are even more things than this project that I presented. So there is a data core. So they are the people responsible for all the data management of that. So I just present, because this is one of 3 projects, what I just showed to you. So there are 3 projects, and there's an admin core and a data core. And that's sort of their duty.
Can I share our experience with this study with this mosquito thingy? No, it's because now we have tried to assess the impact to dengue cases, and we recruited patients. The database was developed by Monash people, the IT from there. But even those who are involved in developing the apps and everything, they cannot have access to the database. So that when there's changes in the apps that they have somewhat need to see the database, they have to request permission from us. So they know that it's really restricted. Even me, I don't have the access to the database. If I request something, I have to go through a database manager, because it's better to have a single access so that every access can be counted, right, and documented better, rather than a lot of people have access and they have access differently. Sometimes it's mixed up things. So if you can have those mechanisms, it's better.
All these app developers always have some kind of a vested commercial interest. Why do they do something? We were discussing yesterday, Diljit and I, about this kind of device. So they may be good, you're the first time, second time, but then we really don't know what they have ideas for future. So I think you need to be very, very careful about your data security.
Thank you for because it's very nice to have all this feedback, because it's a new territory, at least for me. Oh, sorry.
Okay. Any other comment or inputs? Otherwise, big applause. Philippines. You still up to the risk assessment because the data management is still need to work on.
Project that we're working on, we worked with a project that on its earliest stage. So we very much welcome your feedback. It will be very helpful. Okay, so I'll start. It's entitled The Rollout of G6PD Screening and WHO Treatment Guidelines for Malaria Patients in Endemic Areas in the Philippines. To be specific, this is in the Palawan province in the Philippines. So next.
Where is the clicker of that?
Yeah, so the importance of the study is there had been reported cases of induction of acute hemolytic anemia with in G6PD patients or glucose 6-phosphate dehydrogenase deficient patients. And the Philippines Malaria Program has set its goal to eliminate the disease by 2030. However, it is among the very few countries that administer Primaquine drug without prior G6PD deficiency testing. And a study by Dr. Espino showed that the 14% of the population in southern Palawan has G6PD deficiency. So we currently follow the WHO treatment guidelines, but the National Malaria Elimination Program had to adapt it to have lower dosage of Primaquine to address this absence of the screening test.
So, however, lowering the dosage of primaquine from the WHO treatment guidelines may cause relapse of malaria infection in the patients. So that's the problem that we're trying to address. So our research question is, will the use of G6PD screening and WHO treatment guidelines lower the incidence of hemolytic anemia and possible occurrence of relapses in those patients? So the context is, as I said earlier, the Malaria Elimination and Control Program of the Ministry of Health was already done. CHARM had already finished the stakeholders analysis for the use of G6PD screening. And the stakeholders analysis included program managers of G6PD newborn screening, local government units, representatives from private and government hospitals in Palawan. And the conduct of this study was actually recommended by the director of the National Malaria Elimination Program. So that's why it's very fresh, and we have to do it. So yeah, so we're doing this study. And the role and rapport of, I'm pertaining here to CHARM. She's the coordinator of the stakeholders analysis that was recently rolled out. And the rapport that we have with the community is that we've already conducting studies in this field for malaria, and I'm not sure with schisto. I think just malaria, CHARM. Just malaria in the study site, and we have local project staff in the study sites, which was found to be strategic also for community engagement. So we've been there for 10 years already. And the aim of the study is to employ point-of-care testing. The G6PD screening test kit is a rapid diagnostic kit. So it's a point-of-care testing for treatment of malaria, and the use of the actual WHO recommended treatment guidelines in treating malaria patients. So the objectives of our study is to deploy POC testing kits to village health stations and use the WHO recommended treatment guidelines. And specific objective is to determine and compare the number of primaquine-induced hemolytic anemia in both groups. In groups that were not screened and which were treated using the National Malaria Elimination Program, and the other group which will be employed with, wherein we will treat with the WHO treatment guidelines provided that they were screened. And to compare malaria antibodies in both groups, this will show relapse, and to establish the process flow criteria and protocols in carrying out the G6PD screening and treatment of malaria patients. Let me just show our inclusion and exclusion criteria. So all malaria positive patients detected in the village health facility shall be tested using malaria microscopy and shall be recruited except for the following participants: a pregnant woman, those with severe malaria cases, and infants to 2-year-old children. And here are the risks that we identified in conducting the study. First is role enrollment of participants, and mitigation measures include early communication and consultation with community. Risk also on the adverse event, because we will be doing a blood extraction. So we have a number of mitigation measures here. So we need to keep document of vital signs prior to extraction, training of phlebotomists, secure protocol in case of AE during blood extraction, include sexual activity history in case report form to exclude possible pregnant participants, and we need to hire a physician to oversee adherence of guidelines on detection of severe malaria. Also, adverse events due to drug side effects. So to mitigate, we'll do monitoring of study participants, referral to nearby health facilities, and regular orientation of project staff on protocol on handling adverse events. Also, the risk of loss of follow-up due to transfer of address, demise of patient, or lack of capacity to come back due to conflicting tasks. So mitigation measures include researchers to visit participants for follow-up or send text messages to remind schedule of follow-up. Another risk is loss of case report forms in the field. Sorry, typo. A mitigation measure is to scan documents and keep electronic backup, maybe monthly, or you could suggest a more doable time frame. So for a list of documents, before the study, these are the documents that we'd like to keep: study proposal, correspondences, certifications from the village, timeline of activities, the budget, staff documents, CV training logs, and the terms of references. During study, the approved study protocol, information sheets, study briefers, informed consent, case report forms, malaria case forms, lab results forms, SOPs, and directory of contacts. After the study, the list of documents are technical reports, financial report, manuscript, and evaluation results. So there. So we'd like to welcome feedback.
National malaria program treatment will be employed, and then there's no screening test at all, since the current system will not be disturbed or will not be changed. And for the group B, which is the case participants, we will use the treatment guidelines for recommended by WHO, which has 30 milligram of primaquine administered to the patient, and the screening of G6PD, and the baseline as well. And the other one is the baseline for antibodies and hemoglobin count to measure the hemolytic anemia if there is. So that's it.
So in the control group, you will do all these tests.
Yes.
But you will not have any change strategies.
Yes. And also to mention that all incoming patients, and for those that will be positive in malaria testing, will be recruited. So there's no sampling or there's no randomization that will happen. So all incoming patients, all patients that's seeking for treatment or seeking for malaria testing, and if that patient turned out to be positive, malaria positive, she or he will be recruited.
This brings a very big ethical concern. You're not giving them the choice whether they want to be in your study or not, because this is actually a research. Can you turn off your microphone? It disturbs. Yeah. So, I mean, in the control group is justifiable because this is your national standard. But in the other group, it's their choice whether they want to be part of your new or if they want to be part of the existing one. But if you say, no, I don't give you the choice, then where's the question of there being an autonomy being respected? Why did you do that?
That's why that's why we're going to explain and give them informed consent, information sheet, and if they will not willing to participate, and then we will not force them. For those who want to participate, then they will be recruited. But if not, no problem. In their health conditions or health seeking in the particular facility will not be affected at all.
So your inclusion criteria should clearly say participants consented and willing to take part in your study. So it's not only those malaria and whatever that they have consented and are okay to participate in your study. Just include that also in your, yeah?
Okay.
Okay, thank you.
Any more, please? Yeah.
It's just a clarification about the point of care test. Is it something innovation, or actually is it already proved by previous study?
For the point of care G6PD screening, it's already evaluated and it's already commercialized by other countries like Indonesia, Cambodia, all countries that in the face of malaria elimination is already using the POC testing for G6PD. And it has a good evaluation score or rating.
Yeah, but I think that would be very nice to include in the protocol to state that. Yeah. Yeah, we'll include that. Thank you.
Can I?
Sorry, just one clarification I had. was on your question. So, are you trying to show the effectiveness of the strategy, or are you trying to look at implementation of the strategy?
Actually, both. We want to see if how effective the treatment and the screening for the malaria patients. That's why we have the hemoglobin testing and the antibody test, and how they will respond to the new intervention that we will be implemented or that will be carried out in the facility. And also for the health workers that we will do the screening and the administration of treatment.
Yeah, why I ask is because these are two different studies, and combining them into one again raises ethical issues. If you're not sure about effectiveness, do you really want to study implementation already? Because unless something is effective, you don't bring it into practice. So effectiveness is the first stage, and then implementation. But if these are already WHO guidelines which have been tested and you're putting them into place, then your primary question is implementation. And so therefore your focus of study, questioning, outcomes, data collection will all focus towards implementation and not so much towards effectiveness. That's why I just raised there.
Oh yeah, we actually discussed that earlier because we initially had two research questions, but it's a WHO treatment guideline, so I don't think we have to ask whether it's effective or not. We're one of the few countries that don't do G6PD testing. That's why we had to adapt our protocol. So maybe we could just be more clear on our research question that we will no longer.
Yeah, and therefore in your specific objectives also, you're then trying to think through process of implementation, and you're looking at how you're going to assess those and look at those rather than looking at how many have, how many didn't have. So your focus will change by thinking it like that.
Yeah.
Yes.
And just following up from that, because in implementation research, then the intervention is with the health providers, lies in the health providers, but the patient then will be another. So if we talk about in implementation research, there is a coverage. Both you have health providers and also the patient. Now, particularly with the risk, you only see risk from the patient, but the risk of implementation you have to also think through. Yeah.
If this strategy changes, then do they need a control group? If you're looking at implementation and not at effectiveness, maybe then there's no need for a control group, or would you still want to have a control group?
For the control group, we just want to compare the effectivity of the deployment of kits, or how will the BHW or the health provider and the patients will respond to that certain intervention. That's why we need the case and the control group. So we have something to compare with.
The control group then brings a whole lot of responsibility for you. Let's say in the control group you don't do anything, but then they are part of your research. So there's a hemolytic anemia, there's a severe malaria, there's complication. You then have an obligation to be taking care of all this ancillary things, just for the fact that you are using their data. So that's the reason why I asked, do you really need a comparator group, or would you do away with a comparator group? Because you need to think through.
I think there's a design there are several design in IR that we can use that, particularly when we think of also scale up. There's a step wedge design, which you implement like in a step wedge manner. So before you implement in a certain group, it become a control, but later on they will be also included. Yeah.
If I could just add to it, since you will be thinking through design again, and when we do IR tomorrow, it's worth thinking of the use of a control, because a control becomes useful if you already have an implementation in place and you're tweaking it or you're making changes to it, then you want to compare. But where you have nothing, there is no G6PD screening, and you're doing screening, whether you really need control for implementation. So you can think it through of whether you require that design or not.
Okay, thank you.
So applause.
Okay.
Now,
For the methods, we choose mixed method, and we use quantitative and qualitative. For the quantitative one, we do survey, and we also screen the inmates, so we need data about TB suspect, and also secondary data. Tools we use for surveys, questionnaire, and how we ensure the data collected here. We train the data collectors, and we develop SOP, and also piloting the questionnaire, and for the sampling, we to ensure it is whether it is randomized or not. And for data about TB suspect, the tools is standardized symptom screening form. Maybe we can adopt for the standardized one, and we translate it, or we search for the Indonesian version. And the qualitative, we will conduct FGD and in-depth interview. In-depth interview will be conducted between the health staff and also prison staff to assess like their acceptability about this strategy, and whether this strategy will sustain or not in this prison. And the tools for in-depth interview is in-depth interview guidance and audio recorder. The strategies to ensure the quality, we develop SOP and train the trained researchers. So we have to be trained in qualitative data collecting. And for the FGD, we conduct FGD among inmates. It aims is to like their acceptance about these strategies to be conducted in this setting. We use FGD guidance and also audio recorder, and the strategies to ensure the quality is we develop SOP, and we also have to be experienced in qualitative data collection. And we will also conduct the observation here. We use checklists, and we also direct observe them in conducting like the screening. They conduct screening, so we observe whether they do correctly or not. And we use SOP and also our team, research team. So for the data management, we differentiate between quantitative and qualitative. And through this process from the data collection, we differentiate among the process, actors, outcome, and what we will do to minimize the risk. For the process, first is the training data collector. So we have to train the data collector that will do the survey, and it is conducted by the researcher. Outcome is trained data collector, and we minimize the risk by conducting the training by the expert one. If it is IR, so we have to the training will be conducted by the IR people. So for the quality control, after training, and they do data collection, and we do quality control in data collection. It is conducted by our team. The outcome here is we ensure the complete data and risk mitigation. It is random checking, spot check, and for the secondary data, we do like select variable that appropriate for what we want in collecting secondary data. The next one, in the process of data entry, the actors is data entry, and the outcome is field data, and we minimize the risk by like giving the data entry reasonable time to manage, so they do it appropriately. And then we do double entry by data collectors, and data entry checks by researcher. In the data cleaning and data analysis, cleaning validation and analysis conducted by data manager, and the outcome for cleaning is clean data. Risk mitigation we conduct is prioritized variable that will be cleaned. For validation, the outcome is valid data. we do random checking to ensure the quality. And then for data analysis, the outcome is integrated data, and we do, for this one, we ensure the software is original. For the qualitative, it's the same. We differentiate process, actors, and outcome. For the training, it's same. And the collecting data will be done by data collectors, and the outcome is audio recording. Risk mitigation, competence data collectors, and use guidance. And then transcribing, done by data collectors, outcome transcript, and risk mitigation, we train the data collectors because it is done by data collectors. And then the next one is checking transcript, means that after the audio recording being transcribed, then it is checked if it is match or not between the transcript and the audio recording. Data analysis done by the researcher from data familiarization, coding, categorizing, conceptualizing. And you can, for the validation of quality data, we use member checking and peer debriefing to obtain the valid data. For the storing, we differentiate between hard file and soft file. For the hard file, we will keep it in the locked cabinet and can only be accessed by research team and kept for 2 years. For the soft file, it will be encrypted and only be accessed by research team and also kept for 2 years. Thank you.
I think very, very structured. So I like that structured way of presentation.
You want to say? I see your curiosity. No. Franklin.
Just the last bit, you said you store the data for 2 years. Is there a reason for 2 years?
It is only our in our usual practice. It is 2 years.
Just the common practice, but
Yeah.
I mean, if there's any
It's
required
depend.
regulation.
So one of the things they say is also keep it till you have your publication done at the minimum.
Yeah.
So I'm so happy you're so optimistic you'll get it published in 2 years.
I just have a inquiry on this encryption. May I ask how you encrypt your folders?
Actually, for that encryption, maybe it's like password, but no, I'm not an IT expert, so like we will.
Because we had challenges with encrypting folders because it poses a risk. I'm also not a data management person, but we wanted to like use an encrypting software that could be, in case you get locked in, could be, it could be solved. I think there are issues like that. So I thought you have an encryption, so I wanted to gather data here, how you do encryption, because I know it looks simple, but we tried it. We had some challenges with encrypting data. But yeah, it's an IT.
Okay,
issue.
if that's the case, I think you can have like password managers that you put all those password there, and someone have to be responsible.
Yeah, as
to
simple
manage
as that,
Yeah.
right? Yeah. Okay.
Could do is possibly try to look for some links and give you links, so such of you who are interested in knowing further about it, you could read more. Diljit also has an IT background. We could discuss with him and try to find out some good resources, and we provide you with links. Yeah,
Yeah, that
this
would
is
be helpful. Thank you.
I have a question. It's because I'm not very knowledgeable about it, so it's more out of curiosity. So, how do you keep the audit trail when you do a qualitative study? For example, I know there is some flexibility when you make the interviews. So, how you keep track on? Because it's not a survey, right? So you cannot say, "Oh, I made this slight change in the question." How I make it. So, when do you decide, okay, this is an important change in my interview guide, so it needs to be recorded somewhere, and where do you record it, and how, and how do you, because I know it's an iterative process, but I come from a quantitative background, so it's very hard for me to figure out when and how you decide to keep the track changes.
So a substantial change in an interview guide would be like an addition of a new question or a complete change in what the question. Small changes in terms of how you ask them are not considered substantial. So a substantial change is the addition of a new question or deletions, or completely changing the structure of a question. You have to maintain version controls of each of the every time you change it, you make a note in your field notes of why you changed it, and you make sure that version control is there. So that you're using a new version of the interview guide. So the trail is very much maintained. So it's a combination of field notes as well as version numbers. I hope I answered.
I think one of the things that when we do audit trail in qualitative method, whether the researchers, when they decide to switch, has a base and inconsistent. That's the thing when you want to see to do the audit trail. I think that's Ari also.
Yeah, I think in the process of data collection, and then if there is a change of the tools, for instance, qualitative researchers should discuss about that with other researchers before decided that the question is changed or the question is deleted or add a new question. So there should be a process of discussion within the research team, and then the other important things is the content of this discussion should be written in the some kind of notes. So when we have an audit trail, we can show the notes, and then it can be adjusted.
So I think the notes make up a very large part of the qualitative research. The notes is a very substantial part of what you do. Your thoughts, your discussions, changes, everything you note it down. So there's rigorous documentation of everything, and that's how they maintain the audit trail.
Yeah, and just to add to that, I think you're also wondering, so where does the note sit? Like, so where is it, and how are we able to keep track if we want to identify it? Just like the transcripts, when you're using different softwares, you can let it sit there. Like the NVivo allows you to let your notes, all the notes, so the trails, the changes that you have made, they can sit there just like your transcripts are sitting in a database, which can be easily accessed by anybody else so that they can identify the trail that you went through. Yeah.
It looks like a combination of changes you make to your interview guide plus all the documentation that you make in your field notes. That's your starting point, and then from here you then feed it on to the NVivo. So every time you maybe make a major change, then you change your interview guide to say now you added this question, but your field note will say what you did and why you changed your questions. So yeah, for me also this qualitative research is very intriguing. I should do something to really understand what it is. Yeah.
Pertaining to their thing, I just wanted to add two more things. So you do direct observation as well as you are doing interviews. And the reason for this is?
For the interview, we conducted for between the staffs, but the observation, the agents will conduct the screening, so we observe how they conduct the screening.
I get it. And you're also speaking to people to understand the same thing, but why are you doing two different methods?
Yeah. for the validation.
Yeah, so triangulation is the reason you're doing it, and that also is a mechanism of ensuring good quality. So you want to mention it there. You are doing it, but you want to mention that you're going to do triangulation. That was one thing. Second thing is about anonymization within qualitative research, which is quite challenging sometimes. But again, something to keep in mind. How are you going to anonymize qualitative data?
Randomization is in the quantitative.
No, the anonymization of qualitative data.
Dealing with a very sensitive area. Prisons and healthcare providers and prison staff, and that's, I think, the reason why supposed you had something said by a prison staff, and then that puts them into some kind of a risk, no? So then it becomes very, very important in your topic.
Yeah, especially in those kind of setting, people can easily be identified. So that's something you have to be very careful. Yeah.
Thank you.
Okay, so applause. And now we go to Ghana. So we are starting. Okay, so yes, working. So we are starting from the data collection strategy. We had all the methods, the specific methods that we were considering, the three that we mentioned earlier on yesterday, which are the systematic reviews, the interviews, and also the surveys. So, closer? Yes. Yeah, okay, all right, good. So for the systematic reviews, we'll look at the data that we are expecting from the systematic reviews and also the tools that we would want to use. The systematic reviews, we are hoping that we will get both published qualitative and quantitative data, right, on WASH, exactly, on WASH. For the qualitative, our keen interest is in identifying modifying behaviors. So what has previous studies established when it comes to modifying behaviors in relation to WASH? And for that, knowledge is key for us. So those are the things that we'll be looking out for. For the same systematic review, when we get to quantitative, studies that are quantitative, our interest will be in effect sizes. So what are published studies saying, for instance, when it comes to association between the modifying factors and the WASH practices? So these are the things that we'll be looking out for. So how do we look out for these? By using specific tools. It takes us here. So we are looking at coming up with some mesh guidelines that will include some key terminologies that will be used when we are doing the search, okay, or the search strategy. And then we also included the specific search engines that can be used in the search as well. This is just a few. We know that there are several of them. For instance, we use Web Science, Medline, or PubMed, several of them. So they are tools for us. When it comes to the tools, we are also looking at the use of a reference management software that would allow us to do sorting. For instance, we were having discussions with Varal yesterday. When it's likely that you will pick studies through the different search engines that will be repeated across board. So how do you sort when there is double dipping? So that's how we are going to use some of these softwares to allow us to do the sorting and categorization at that level. For the interviews, when you get to the interviews, we have a number of categories of people to capture what? Knowledge, attitudes, and perceptions when it comes to some of these WASH practices that we are interested in. And for each category, we would have an interview guide. So if it is for the food vendors, we'll have an interview guide that specifically will be asking questions that is relevant to the work that they do. And we'll do same for all the others. However, in addition to that, our field notes will be key, as has been mentioned, and also our recorders that we'll be using. When we come to the surveys, we'll be doing just household heads. They will be our interest. Why? Because when it comes to the WASH intervention, it is the environment within the home that is likely to enable the picking up of these behaviors or practices. So it's the household heads, and it's basically going to be quantitative. What do we do? As we mentioned earlier on, this is going to be informed by this and that. So our survey development will be informed by the information that we get through the systematic reviews and the interviews. We would use that just to survey, to identify how we can put knowledge and watch practices together. How are they associated in practice? And then we would also need our neighbors. And then we'll only have questionnaire that will be for the household heads. Okay. Now moving on to the data management plan. So again, we will do it systematically for each of the designs that we have. So for the systematic review, we were looking at capturing the information under the data collection processes, one, the data collation processes, which would involve the management process, as well as the quality assurance processes. So what do we put in place to put checks in? So for the systematic review, we are starting with the identification of the databases that would be used, the search strategy, including the mesh guidelines that we'll come up with. How are we will be doing the sorting and the categorization with the Mendeley, so it's just feeding into this process. At the collation level, we move into how we will be managing the entire data that we would collect through the process. So for the first thing, we say that data will be stored in the University of Ghana OneDrive folder. Franklin has something to say about that when the questions are coming. The selection of the studies will be based on pre-specified inclusion and exclusion criteria. For instance, it's not every study that will be included. We'll only be interested in looking at diarrhea-related diseases, studies that are associated with diarrhea-related diseases. And what quality checks do we put in place at this stage? We are saying that selection will be conducted independently by 2 raters. So an independent rater is here, the other person is there. However, whenever there are disagreements, they will be resolved through discussion. And then we also do what quality will be assessed using the Critical Appraisal Skills Program checklist. It's a tool that is available to help us to ensure quality for the systematic review process. For interviews, the processes are a little different. We start with the purposive selection of the participants. For example, the food vendors. We wanted to use the food vendors here for a number of reasons. Unlike the head, we would use the household heads for the survey. We'll conduct and record our interviews. We'll take field notes and any other thing that is of interest there. Then when you get to the collation, we are talking about transcription, familiarization with data, coding and categorization using NVivo. Again, we'll store data here and then the data interpretation processes will go on. For quality assurance, we'll do member checks. One, we'll also establish credibility via respondent validation through close working and sharing of research findings with relevant health professionals. So that is the part of the validation processes that we'll be engaging in. We'll also do dependability via standardized approach to data collection and audit processes. That is for the interviews. And then for the survey, similar processes, but what I want to emphasize here for the surveys, which is a little different, is the mapping out that we'll be doing within the hotspots. So these hotspots, as we mentioned, are densely populated, but we say that we will be identifying household heads. How do you identify them? Those are going to be very intense processes, so we need to do a mapping of these densely populated areas to be able to identify the households ahead of time. In Ghana, we call something the enriching areas data that we would have to access through the Ghana Statistical Survey Department to do this mapping before we move out. And we look at that as a key part of the data collection processes. Then we will do paper-based interviewer-administered questionnaires, clarified. So we'll do our data entry, we'll do our storage, we'll lock in cabinets, we'll do data analysis. When it comes to the data quality, we are saying that we'll do double entry verification, data cleaning, and then we'll do creation of the dummy tables that will allow us to perceive how we want the data to be presented. That will help us to go back and forth. If we are not getting what we need, then we make sure that we streamline it accordingly. Thank you.
Thank you.
We are taking notes.
Thank you for your presentation.
First and foremost, we try to anonymize the data. And we are trying not to get too worried because the data is anonymized, and the University of Ghana has quite a tight IT service that would ensure that the data is not easily breached. Yes. So as you indicated previously, so accessing the data will have to go through. We would have a manager who would then liaise with the computing service. So anybody who have access to the data will have to go through the same person.
Survey data, is it quantitative data?
Yes. Yes, it is quantitative data.
I'm just having difficulty understanding what kind of data would you be asking in the survey?
In the survey? Oh. Oh, okay, I think we missed that. Even in there. Yeah. Okay, so knowledge and watch practices. So it's going to be quantitative because we want to do associations and find out how knowledge is associated with the watch practices. Yeah, so it's going to be basically quantitative data. Yeah. Any more? Yes.
So regarding the systematic review, I don't know if you also have considered software for managing the review itself, because if you get very few papers, then it's okay, but you may end up with thousands or hundreds of papers. So I think even more than the Mendeley, I mean, the reference manager is one thing, but also for managing who decided what criteria, what was used for excluding, all these things also should be, so there are softwares for managing that.
Okay, all right. For example, which ones would you suggest?
So there's one that's called Covidence. It's from the Cochrane collaboration. Okay, I think you can do up to one for free, and then after that you have to pay. But
like up to one analysis, like a whole study, and beyond that.
We have used it. It's very good, so because you can, for example, track the changes because you decided to change, and you make the. So it has what each reviewer have decided, and what the agreement was. You can export the data into also analysis if you want to make analysis in other softwares. So. And you can access it later. Mm-hmm.
There's another one called RevMan, Review Manager. That's also, it's free, I think. I don't think you have to pay for it, but that's also quite effective. It helps you to do quality appraisal as well within the system.
Okay, all right.
When you do a systematic literature review, you have a standard called Prisma.
Prisma.
Yes. Yeah, so if you want to make a publication of your, I see you can make multiple manuscripts at the end as well. So if you're looking at just publishing your systematic literature review, then you need to follow the Prisma guidelines. Okay. This is also available, and it gives you, I think it's very good because it then enhances the quality of your systematic literature review itself.
Yeah, since we're on the subject of systematic review, just wanted to add one or two other things. So the process for a systematic review is quite well laid out and specified in terms of steps that need to be done, things that need to be reported. It's all there in literature. Very good guidelines are there. In your data collection tools, I think the biggest data collection tool for a systematic review is the data extraction sheet, and I don't see that.
Yeah.
So that needs to come in. And I was also just wondering, again, just clarification of the need for a systematic review. I mean, have you looked at literature already? Aren't there enough systematic reviews on WASH practices, and or is it very little? I don't know. Just checking. Because again, that raises an ethical concern of putting resource into doing a systematic review if there are already enough done.
Just to say this, that it's a big team, and we have like 2 experts on the team doing the systematic review. That's why we are not able to answer all the questions as you expect. But they found the need for it. Through their own experience, they thought that it would be good for us to establish it, especially because we want to build the behavioral change strategy, a new intervention to put in place. We need to understand what is there already. Yeah.
Sorry for extending the systematic review discussion. I think after this we can stop it. But also whether you want to think of then separating maybe low and middle income countries or only Africa, because these are contextual issues, especially qualitative when you look at. And if you are going to derive and base your interventions on it, then you want to try to limit and not make it global. Yeah.
Exactly. Good point. Yeah.
Okay.
Thank you very
Any
much.
more?
Any more?
No more?
Yeah. I think we still have one participant, and we